And today we have Antonia Massmann here. She is working at King's College London together with Tom Arici. And I think a lot of people at ISMRM also saw her work on high resolution layer dependent fMRI with her robot, specifically to trigger a very specific responses in the somatosensory cortex. And she's also working on infants, and her title today is The Development of Sensory Processing in the Human Infant Brain. Yeah.
Thank you so much for the introduction, Renzo, and thank you so much for giving me the opportunity to give this presentation. Yeah, I'm a second year PhD student at King's College London. I have a neuroscience background, and I'm just going to present a few things that I've been doing so far. Okay, so first of all, why am I interested in the neonatal brain and in neonatal brain development? So this is because the neonatal period is one where we see lots of changes happening in the brain. So already in the weeks leading up to birth, so in the last trimester, as well as in the weeks post-birth, the brain is rapidly changing in structure as well as function. And we can observe this at a global as well as a microscopic level. So whole brain regions are forming and connections between them are forming. And then we also have the maturation of individual neurons and so on. Any abnormalities during that time period, as well as, for example, pathologies, can have lifelong implications. And we can see this, for example, in neurodevelopmental conditions like autism. So to give you a few examples of what's happening during that time period, we can here see a brain at 27 weeks postmenstrual age all the way to 37 weeks postmenstrual age. And this is roughly the time of birth. And you can see that in the 10 weeks leading up to birth, the brain is, or the cortex, undergoing the majority of its folding. So you can imagine how many changes are happening during that very short span of time. At the same time, our cortical layers are forming as well. So at 27 weeks, we can still see the cortical plate as well as the subplate. And then over the next 10 weeks, the 6 layers that we're quite familiar with when we start talking about layer-fMRI are being established. Simultaneously, we also have the migration of neurons. So the important in and outputs are starting to be established and migrate from the subplate all the way up to the cortex, so that at the time of birth, we have the vital in and outputs established so we can start interacting with our environment. Apart from these neural changes, it's important to mention that we also have lots of changes in the physiology. And when we talk about fMRI, it's important to mention that this is also, or includes the microvasculature as well as the neurovascular coupling. So around the time of birth is only when our cortical capillary network starts developing. So as you can see, quite rapidly, we have lots of changes in vessels as well as the metabolic demand. So talking about sensory processing and sensory development, you can imagine that with the brain changing as much as it is, this is also something that's constantly changing. So in the adult, when we talk about sensory motor processing, you can see that the majority of our inputs are relayed from the spinal cord by the thalamus to their relevant primary sensory cortices, in this case, the somatosensory cortex, which projects it onto associative cortices, or in this case, the motor cortex, which then feeds it back to the periphery and some subcortical areas and so on. So this very complex global network exists. And in the infant, that is still developing, with each of these different sensory systems and sensory cortices, which then projected onto other areas, also having their own developmental trajectories. You can see this here by looking at different plasticity windows across postnatal days, so days post-birth, and you can see that different sensory systems have different windows in which we can see rapid changes in their development. In my PhD, I'm focusing on 2 specific sensory domains. Those are touch and vision. Touch is currently widely understudied, despite its importance in infancy. We need it, for example, for accurate cognitive development, like interoception, but also for complex social skills like communication, emotional regulation, and attachment. You can see that the relevant developmental windows, which are the somatosensory as well as the insular, are much earlier compared to the visual system that I'm also looking at. The visual system's developmental window is the last to open and the most protracted, and early visual inputs are very vital for the maturation of our visual function to facilitate an accurate representation of our visual field. So any disruptions during that time period cannot be recovered later on. And that means that it can affect things like social behavior, so we may not be able to recognize faces as well. So therefore, I'm looking at these two sensory systems, and going forward, I'm first going to talk about the visual experiment that I've been doing. This is something that I've been doing in collaboration with Future. So how much can a baby actually see when they're born? As you can tell, not that much. Everything is mainly black and white, lacking contrast and definition. However, the adult visual system is able to perceive the world with all of its colors and details. And so you can imagine how much functional and structural development needs to happen in order for our visual system to be able to do this. We can illustrate this by looking at the structural changes. So here we have a neonatal primary visual cortex, or V1, and you can see that the majority of these important cortical and subcortical in and outputs, including the important thalamic inputs from the LGN, which reach the middle layers, they are fundamentally established, however, not fully mature yet. But we do have some mature short cortical-cortical connections. Over the next 10 to 14 years, this neonatal adult V1 configuration matures into, or neonatal configuration matures into that of an adult. So now we have very mature connections, still including the important thalamic inputs from the LGN to the middle layers, but we also now have connections across layers as well as to extrastriatal areas like V2. So there's lots of functional as well as structural change and development that needs to happen for our visual system to be fully functional. But our visual system isn't just V1. In fact, there's this theory that different components of our visual environment are processed by two distinct streams. So we have the dorsal stream, which carries information about spatial location, motion, and low contrast stimuli. And then we also have the ventral stream, which is important for processing fine detail, texture, color, and high contrast stimuli. This was first discovered by Haxby et al. In 1991, and they used a PET study where they gave participants either a face matching task or they had to match the location of a dot relative to a line. And what they saw was that very reliably across all of their participants, when they had to do this location matching task, they saw this increased blood flow in the superior parietal cortex, which became the dorsal stream, whereas the face matching predominantly engaged the occipital temporal cortex, which became the ventral stream. Developmentally speaking, these two streams are thought to have different trajectories. The dorsal stream appears to be particularly vulnerable in early life and is implicated with a number of conditions in preterm birth, but we don't fully understand this yet. However, because these streams are distinguished by their tuning to contrast and temporal frequency, we can, by varying these stimulus parameters, preferentially engage one stream over the other and therefore start to probe their functional development in this critical period. To do this, we need to use ultra-high field fMRI because we want to gain an insight into the underlying functional circuitry. And because of the submillimeter resolution that we can achieve with 7T, we're able to probe differences in the bulb response across cortical depths, like it was done here by Polimeni et al. In 2010, which showed this altered representation of this M stimulus all the way from the peel surface down to the white matter in the adult V1. We can use these depth-dependent characteristics to then interrogate the functional circuitry of the cortex. So, for example, in this visual working memory task, they inferred a cortical circuitry model of V1 and V2 with these feed-forward and feedback in and outputs based on their laminar-specific activation during the task. So this brings me to our aim, which was to test whether stream-specific processing can already be distinguished in the neonatal brain across cortical depth by varying the contrast and temporal frequency using ultra-high field fMRI. To do this, we created 3 different visual stimuli, which we presented in a block paradigm of roughly 18 seconds on and 28.5 seconds off. So we had these flickering visual stimuli presented with black screen off periods. Stimulus 1 was this high-frequency, low-contrast stimulus, which was meant to preferentially activate the dorsal stream. I really hope that you can see the flickering correctly. Sometimes with streaming, it gets really laggy, but you have the proper stimulus parameters here in case you're interested. Okay, so stimulus 2 was a low-frequency, high-contrast stimulus, and that was meant to preferentially engage the ventral stream. And then lastly, we also had stimulus 3, which was high frequency and high contrast. So that was not meant to be stream specific. After we've developed those three stimuli, we started collecting 2D gradient echo API data on the 17 magneton TeraX scanner with a resolution of 0.8 millimeter isotropics and a TR of 2.59 seconds. All fMRI images that we acquired were with a restricted field of view, and you can see this here in blue. The data I'm going to show you is from 9 healthy neonates. They had a mean age of 39.6 weeks post-menstrual age, so that is the time of typical birth. But we collected data all the way from 33.5 weeks, which is a very preterm baby, so a baby that was born very early on, all the way up to 46.5 weeks. So there's a baby quite late post the typical time of birth. They were all sleeping, so they had their eyes closed, and we used a 1Tx head coil because of local safety assessments based on simulations. We had a TE of 50 milliseconds, acquired 50 slices, and had an SMS and a graph factor of 2. Overall, we used more conservative style limits and had continuous monitoring of temperature and vital signs during the entire acquisition to ensure the safety of our infants. We also collected data from 6 healthy adults. They were awake and had their eyes closed. Here we used a PTX coil. We had a TE of 25 milliseconds, acquired 72 slices, had an SMS of 3, and a graph factor of 4. When it came to our data analysis, we first pre-processed the data by doing thermal denoising with Nordic. We did motion correction and then ICA denoising on our neonatal participants and excluded any that had excessive motion before extracting clusters of activation using GLMs. We started the analysis by defining ROIs in V1, and that's also the data that I'm going to show you because we wanted to see how these different stimuli are represented at this very early cortical entry point to the visual streams. And we define that ROI based on the neonatal bounty atlas or the adult pre-surface segmentation. We delineated the ROI into 3 cortical depths using NANI and then extracted the depth-dependent time series. So before I'm going to show you the fMRI data, I just wanted to briefly highlight the structural images that we also acquire across all of our neonates. And so I picked 3. So one for within the preterm age, so with early birth, one at term age, so like the standard time of birth, and then one all-term baby, so that's a few weeks post the typical age of birth. And I hope you can appreciate all of the developmental changes that I mentioned at the beginning. So you can see the drastic changes in gyrification in the cortex. We can see changes in the contrast, for example, in the white matter. And it just really goes to show that we can use fMRI to capture these developmental differences and changes across that very short span of time. Okay, so when we looked at the fMRI data, in response to all of our visual stimuli, we could see a positive bold response in all of our infants below 42 weeks postmenstrual age. So here we can see that this is consistent across the entire acquisition period. And in this exemplary infant, we can see that it's nicely localized towards the visual areas, and we can even see some activation of the LGN. In our adult participants, we similarly saw a positive bold response localized to our visual system, but here that was quite reassuring for us, to be honest. We could see some spatial differences in these activation maps. So for example, stimulus 2 had this more lateralized response, which was quite reassuring because that was the stimulus that was meant to activate the ventral stream, so the more occipital temporal areas of the cortex. And even by looking at the time series in the adults, you can see some differences across these stimuli. So stimulus 3 seems to have a larger response amplitude compared to stimulus 1 and 2. We then looked at these responses across cortical depths in V1, and in response to stimulus 1, which was the high frequency, low contrast stimulus, we could see this delayed onset in the response compared to the adults, with a peak of around 5%, roughly 20 seconds post-stimulus onset in the superficial depth, before the response then slowly returned back to baseline. In the adults, however, as I mentioned, we had this faster onset, a sustained or prolonged response towards the stimulus with a second bump before it then quite sharply returned back to baseline. In response to stimulus 2, which was the low frequency, high contrast stimulus, again, we had this delayed onset in our neonates, and then we saw this overall reduced response amplitude to the stimulus before then the return to baseline, and we could observe this negative post-stimulus undershoot in our neonatal participants. In response to stimulus 3, which was the high frequency, high contrast stimulus, after the delayed onset, we again had a peak of roughly 5% in the superficial depth, which was much earlier compared to stimulus 1. So here we could roughly observe it around 14 seconds post-stimulus onset. The response then returned to baseline, and here we were also able to observe this negative post-stimulus undershoot. Since our stimuli share different properties with one another, we were able to tease out two potential effects. So stimulus 1 shares the high frequency with stimulus 3, and in both cases we could see this larger response amplitude, whereas stimulus 2 and 3 share this high contrast property, and in both cases where we could see this negative post-stimulus undershoot. So to summarize, high frequency seemed to drive this larger response amplitude in our neonatal participants, whereas high contrast seemed to drive this negative post-stimulus undershoot, which might be due to high metabolic demand in response to this high contrast component of the stimuli. And overall, this suggests that the neonatal V1 may already be able to encode frequency discrimination and luminance contrast, which might perhaps facilitate the development of the stimulus-specific processing across the visual streams later on. Overall, the neonatal responses were slower, so they had a delayed peak at around 15 to 20 seconds and a delayed return to baseline around 28 to 32 seconds. Adult responses, on the other hand, rose much faster. They plateaued throughout the stimulus presentation and then returned sharply back to the baseline at around 24 to 26 seconds post-stimulus onset. So then I took the peak responses to these stimuli and just averaged them and looked at how do they differ across these three stimuli from the deep all the way to the superficial depth in neonates as well as adults. And overall, we can see that in response to stimulus 3, we have the largest response amplitude across all depths, which may not be surprising since that is the stimulus with the higher frequency and the high contrast. However, in the neonates, that was followed by stimulus 1, which had the higher frequency, and then by stimulus 2, which had the high contrast, whereas in adults, those two seem to be pretty on par. So this again kind of supports this notion that maybe in the neonatal V1, we have some form of encoding of these different stimulus features that we can't really observe in the adult cortex. So another thing and somewhat surprising result that we had was that we also did this in older infants. So I just mentioned that all of these results were in neonates below 42 weeks, but we also collected data in neonates that were older. And here we could actually see that with increased age, the response amplitudes became increasingly negative, and this was observable across all three stimuli. We still need to understand this a bit further. Yucha is actually looking into this. We have collected a lot more data, so we'll see what those results say. But at the moment, we think maybe that is reflecting a developmental change in the hemodynamic response. It could be related to a growth of the inhibitory circuitry during that time, or be linked to altered sleep stages, which may therefore cause differences in arousal. Or it could be due to an increased neural density, and therefore greater metabolic demands, which cannot be met by a slower maturing vascular system, which may result in that negative bulb response. But it's definitely something that we're trying to understand in more detail. So to summarize, we can use ultra-high field fMRI to investigate visual processing of different visual stimuli in the neonatal period. And we have shown that while the deep to superficial increase in amplitude for different stimuli is comparable across neonates and adults, the temporal dynamics differ, which suggests that the neonatal V1 may already encode frequency discrimination and luminance contrast at birth, which indicates or could be indicating that the foundational visual circuitry is already established at this early cortical entry point of these visual streams, and which might be a way of supporting the further stimulus-specific processing across them. By extending the analysis to higher order visual areas, it will allow us to investigate how this visual pathway development from the onset of and throughout this critical period is progressing, including in preterm infants, which is this population of a heightened risk of visual impairment. So, as I've introduced in the beginning, the visual system is only one of the sensory systems that I'm focusing on in my PhD. The other one is touch. And the reason why I'm looking at touch, and specifically in neonates, is because in infants, it's actually crucial for cognitive development as well as for complex social skills. It was also found to be important for pain reduction. So, gentle tactile stimulation has shown to lower noxious evoked brain activity during clinical procedures. It's shown to enhance infants' attention and face identity recognition, which supports caregiver attachment, as well as help with stress regulation. So it lowers the heart rate and the autonomic arousal in preterm infants. But when we start talking about touch, we need to start in the periphery. And in the periphery, we have two different types of skin. So human skin is often categorized in hairy or glabrous, and each is densely innervated by a number of specialized sets of receptors and nerve endings, which are tuned to different qualities of touch. In hairy skin, we have these fast-conducting A beta fibers, and they're thought to carry discriminative information to the brain, which are things like poking, vibrations, or faster stroking on the skin, so above 10 centimeters per second. We also have these unmyelinated CT fibers. These are slow conducting and are tuned specifically to gentle and slow stroking at around 1 to 10 centimeters per second. These CT fibers are thought to underpin the affective or emotional component of touch. So that's including things like pleasantness of being stroked, the social bonding, and the caregiving, which are the components that are very crucial for these infants. Effective and discriminative touch are thought to be carried to the brain by two distinct peripheral pathways before they start converging into their central processing hubs, which includes the insula as well as S1. However, even at this cortical level, the segregation of these two streams is thought to continue. So in S1, the discriminative inputs from the A beta fibers are thought to terminate predominantly in the middle layers, whereas effective and contextual signals from the CT fibers, which includes feedback from the insula, are thought to predominantly arrive in the superficial and the deeper depths. From these central processing hubs, each stream feeds into its own higher order processing network. There have been fMRI studies on this, and they try to look at with manual stimulation. So that's basically someone who's standing by the ball during the acquisition and manually, for example, brushing a participant while they're in the scanner. And with this manual stimulation, they have done a few fMRI studies to look at this pathway, which was predominantly established through animal studies. And they have shown that in adults, you can see these global blobs of activation in the primary somatosensory cortex as well as the posterior insular cortex. Similarly to these adult studies, they have also shown a posterior insular response and a post-central gyrus, so an S1 response, in neonates 1 week post-birth. However, these 3D studies, 3T studies are we're only able to see these global activation maps. We're not able to functionally probe its underlying circuitry, which is why we want to do this in an ultra-high field fMRI scanner. However, in order to do that, we need to deliver a reliable stimulus. And as I mentioned, the previous studies have been using a manual stimulation, which for multiple reasons is very difficult to do in a 7T, which is why the first thing that we wanted to do was to design and validate an optimized system to generate a controlled, effective tactile stimulation in the 7T, and to then use this to explore the underlying brain processing with ultra-high field fMRI. All of the robotics that I'm going to show you now that we developed for this were done in adults because we wanted to do this first to actually properly validate it before we start using it in neonates and work on a neonatal version of it. So the adult version had 5 core requirements. It first of all had to deliver a reproducible stimulus at controllable speeds. It needed to be easy to set up and control. It had to be fully MR compatible, be low cost, and also have this modular design so that we could use it in our infants. The solution to that was this pneumatic design or system, which consists of this laser cut polyactic base that is supporting this 3D printed arm, which has in yellow, as you can see here, a piston attached to it. So in the piston, air pressure accumulates or decreases, which moves this carbon fiber rod by extending or retracting it. On the end of that carbon fiber rod, we have another 3D printed piece which holds another carbon fiber rod. On that end, we have either a 3 by 2 centimeter effector, which is gently delivering a tactile stimulation on the participant's skin. Or on the other end, we have this fiber optic implementation, which gives us insight into the rod's displacement. The whole setup looks like this. So we have a laptop which sends commands to an MRX1 pneumatic controller. Both of these are outside of the scanner room. And this MRX1 pneumatic controller then pushes air through long tubes into the bore. And inside that bore, the air actuates this MR compatible end effector, which then moves across the participant's right forearm at a programmed speed. Crucially here, no electronics or ferromagnetic components go anywhere near the magnet, and we can read out the actual movement of the device using the fiber optics that we implemented. This is what they look like. So essentially, we have this reflective stripe pattern that the fiber optic shines on, the light shines on that. And then depending on the reflective pattern, we can read that out, which looks like this. So here's the raw fiber optic signal. And as you can see, we use a block design. And as you can tell, we get this very clean periodic signal that has no MR-induced artifacts. And when we zoom into that, you can even see those stripe transitions within a block from the movement. We can then use these transitions and calculate the speed of the device. So that's what it, for example, can look like. So here we have a reliable stimulation above the CT range. So that is above 10 centimeters per second. And you can see we have very low variability within a block, and it's reliable across the entire simulation period. What's even nicer is that we were also able to generate a slow stimulus with that same device, so within the CT range, so between 1 to 10 centimeters per second. So overall, we were able to use this device to produce 2 reducible stimuli, one within and one outside the CT range. And both of these were reliable and reproducible across blocks, within blocks, and also across participants. So once we've done that, we started collecting data with it. So essentially, we just strap it onto the right forearm of our participants. We make sure it's horizontal as possible. So we prop up the elbow and make sure that they are comfortable to be in that position for the whole duration of the scan. We collected data from 7 healthy adults in total. So we collected 2D gradient echo API data again on our 17 magneton TeraX scanner. We had a resolution of 0.8 millimeter isotropics. And we first collected data from 3 healthy adults with a partial field of view. And we did this because we specifically wanted to target S1. In all of these cases, we used the PTX coil. We had a TR of 2.59 seconds, acquired 72 slices, had an SMS of 2, a graph factor of 4, and an echo time of 25 milliseconds. Afterwards, we also collected data from 4 adults using a whole brain field of view. Here, however, we had a larger TR, so it was 2.99 seconds. We acquired 120 slices and had an SMS of 3. And the reason why we switched this is because we also wanted to get some data from the insula instead of just S1. During the scan, we did two separate acquisitions. So we did one within the CT range of 1 to 10 centimeters per second, and then one outside the CT range, so above 10 centimeters per second. We didn't do this in a particular order, so this was pseudo-randomized across participants. But in both cases, we had a block design of 10 TRs off and 10 TRs on, so roughly 30 seconds on and 30 seconds off. We pre-processed the data using thermal denoising, motion correction, and extracted clusters of activation using GLMs. Afterwards, we defined our RIs in S1, first of all, based off of the adult free surfer post-central sulcus segmentation, but we also made sure that it was medial to the hand knob where we could see these clusters of bold activation. Afterwards, we used Laney to split the RI into 10 depths, applied linear deveining before extracting the depth-dependent time series. So what did we find? In response to both of our stimulus conditions, we could see this robust activation in S1 in kind of the expected somatotopic region. In the left here, you can see the CT fiber response, and on the right, you can see the non-CT fiber response. While these two are in the expected somatotopic region, we can see some differences in the spatial extent and in the pattern across these two conditions. In the insula, we could also see a clear activation in response to both stimuli. What was really nice to see here and quite reassuring is that specific to the CT range stimulation, we have this activation in the posterior insular cortex, which was also bilateral. And that is consistent with the literature in terms of where we would expect CT afferent inputs to reach the insular cortex. Similarly, in response to the non-CT range stimulus, with this more anterior insular activation, which is also consistent with the literature. So at the whole region level, the device is doing exactly what we designed it to do. It's dissociating effective from discriminative processing. We then looked at this across the cortical depth. So we extracted the depth-dependent time series in S1 around the central sulcus. And looking at the depth resolve profile, you can clearly see a difference across these two stimuli. So the S1, the CT fiber response is in black here, whereas the non-CT fiber response is in gray. And when we look at this across time, we can see that in response to the CT range stimulus, we have this largest response amplitude in the superficial layers or depth, which is consistent with the idea that feedback inputs are entering as part of the effective processing stream. In response to the non-CT range stimulation, we have a different tempo and amplitude profile with a stronger engagement of the middle layers relative to this temporally delayed or these temporally delayed and reduced superficial response. This is also consistent with the literature, which has this idea that the feed-forward A-beta thalamocortical inputs are reaching the middle layers of S1. So for the first time, we can see that the two touch pathways are differentially expressed within S1 itself in vivo in humans at the level of cortical layers. So to summarize, we've built a pneumatic MR-compatible robot that reliably delivers an effective and discriminative tactile stimulation in the ultra-high-field fMRI. It enables us to investigate the cortical circuitry of touch at the laminar scale, and we have shown that CT range and non-CT range processing engages distinct depth profiles in S1, which is consistent with the literature. Because the design is modular, it adapts easily to other populations, and which is why we want to do this in critical developmental periods like the neonatal one. So overall, with the visual experiment, we have demonstrated that stimulus-specific encoding seems to already be in place in the neonatal V1, and by expanding the laminar analysis to higher order visual areas, we can start to decipher the functional development of this stream-specific processing in the neonatal period. When it comes to touch, we have shown that consistent with the current literature, which is mainly based on animal studies, the CT and non-CT touch pathways are differentially expressed in the adult S1. And by finishing the development of the neonatal touch robot, we can use this to characterize the laminar response to CT and non-CT range stimulations and how these two touch pathways are functionally emerging in the neonatal period compared to the mature adult brain. So overall, we have shown that ultra-high field layer-fMRI is a powerful tool to probe the development of sensory discrimination in this critical period, with the potential to understand and characterize early disruptions which are implemented in neurodevelopmental conditions like autism. With that, thank you so much for everyone who's involved in my project, in particular my supervisors, Tom and Nick, as well as Yucha, who's a vital help when it comes to all of the laminar fMRI, and Ilda when it comes to the robotic development.
Thanks a lot. That's really cool work, and then quite a lot already. Yeah, for these years of the PhD. Maybe I can start with the first question, and then I'll ask around. And maybe the first one is about the delivery of the visual stimulation on sleeping neonates. So how is that done? Are you kind of illuminating the entire bore, or are you using mirror? Yes.
Yeah, so we basically, because the stimulus is quite simple, so it's just a flicker. And the screen is extremely bright. So we've all done it and tested it, whether we were able to see it in the bore, even if we weren't perfectly positioned, because with infants, they obviously move quite a bit. And we also use an adult head coil. So can they see the mirrors perfectly? Probably not. But because this room is always dark during the acquisition, when we actually show the stimulus, it is illuminating the whole room, including the bore, which is how we're able to get these visual responses.
Cool. So you use a screen and a mirror. Oh, cool.
Yeah.
Is it the same then for the adults, that they have their eyes closed and you're, yeah, okay.
Yeah. Yeah, we always say, please keep your eyes closed. If you fall asleep, that's not a problem because that's what the babies do. But yeah, make sure you don't squint too much because obviously adults have thicker eyelids, so there's potentially less light going through them. But yeah, they always have their eyes closed as well.
Cool. Are there any questions? Maybe I can ask another one on the visual stuff.
Mhm.
So one intriguing thing that you showed was, I guess, the more lateral response also for some of the frequencies, for the high frequency with high frequency with low contrast. That reminded me of a study that I found from, I think, Camille Uluda, where kind of he showed that when you don't use a canonical HRF, and you only use the transients, that you get different areas, like the more lateral ones, as opposed to the center of the usual only V1 areas. So you see that in neonates, the HRF is generally slower. So you don't really have these kind of transients as pronounced as in adults, I guess. Did you play around with different HRFs, or do you think that, I guess, the reason why you have this lateral response in adults and not in infants could be due to due to this different sensitivity of the vasculature to these transient time points? Mm.
Yeah, I think that is a really good idea. I mean, I use age-appropriate HRF. So Tom in his PhD characterized the HRF for a preterm and term infants. And so that's what I use to input into the GLM. And then if we have babies, for example, which are at the cusp of preterm or term, I do try both just to see how robust is the response, do I see differences in the activation maps, and so on. I haven't, yeah. So I do that, and then I always look at negative fold as well. So I look at the inverted version and put that into the model to just see what we can pick up. I need to do like a proper group level analysis and actually look at the activation maps across different ages, as well as yeah, post-menstrual age. but also postnatal age. So do we see differences the longer they're born and the more input they have, versus just age post-conception? But yeah, that's something that I'm currently working on.
Cool. So I think Wietske was the first one to raise their hands, and then Farouk.
Yay, you get to push ahead. Thanks for the talk also. Yeah, this is more of a follow-up question to this answer and also Sylvia's question in the chat. As wondering if you could talk a bit more about those, like you can ask your own question, Sylvia. Don't be. But those negative bold responses, like because adults don't have this, right? So what do you think is happening there? Like why did they go negative? I don't.
Yeah, that is a very good question and one that people have been asking for a while because the earliest visual experiments they showed a negative bold response. So early visual experiments with 1.5 Tesla, but also 3T, especially in older infants, have shown this negative response. And then in the literature, some participants or some people report positive responses, others see negative. And we're not entirely sure when we started doing the 7T experiments. The reason why we did that is to see if maybe both is present, but just because of the lack of spatial resolution at 1.5 and 7T, it kind of merges into one blob, which is why we either see a positive or negative. But with the 7T at the beginning, we could see predominantly just positive responses, which was quite reassuring. But then when we started looking at these older infants, we could see a negative response as well, which is why we were quite surprised. And yeah, this is what it looks like. And we're not entirely sure why this is happening. We did have a few ideas. This could be because the brain obviously is massively changing over the time, like over these few weeks. So if new neural populations, or we have this increased neural density, whether that's inhibitory or excitatory, it might mean that we have a greater metabolic demand. But since the vascular system is slower in its maturation, it might mean that it cannot be fully met, which could result in this negative bold response. Could be due to altered sleep stages, because again, sleep is something that's rapidly changing during that time period. And there have been a few studies that have shown that in adults while they're sleeping, they get the suppressed activation in the visual system. So it could be that maybe the sleep is more mature in older infants, therefore we get kind of the suppressed activation. But again, we're not entirely sure, and this is why we've collected a lot more data and we're trying to.
Yeah. So I guess I've never tried to scan sleeping people, whether kids or like adults. So does it like would adults have a negative bold response to a visual stimulus if they were asleep in the way that the infants are asleep? Or do you know that?
So, and across different sleep stages, they have seen that suppressed response. So N1 and N2 sleep, if I remember correctly. The thing is with our infants, we don't sleep stage them while they're in the scanner because you need more sophisticated equipment. So this was only done in studies where they, for example, had EEG or simultaneous EEG as well. We can't do that. So it's really hard for us to actually tell what sleep stage they're in, how deep they're sleeping. We know that they are sleeping, but we're not really sure what kind of sleep it is. So it's
No.
quite
And you
hard.
can't scan them awake, I guess.
Oh, I mean, with the infants, it's just too much movement. Especially, I guess, with the visual stimulus as well. I can't imagine that they would particularly enjoy lying still.
No, probably not. No. Right, thanks.
Cool. Sorry, Sylvia, for missing your question in the chat. And maybe this also
No
responds.
worries.
This answered it, right? Yeah.
Yep.
Cool. And then Farouk, do you want to unmute?
Yes. Hello. Nice talk. Thank you. First, just a follow-up like to Witzke. There is negative bold response in adults. I saw it in many visual experiments. It's just that they are in fewer in voxels compared to the positive responding voxels.
Yeah, but they're not in this area in V1, right?
No,
They're in
there is
like what
there is
represents to the outer visual field or like
No, I don't think so. They are here as well. Like, I mean, I think there will be something coming out soon showing actually there are negative responding voxels around this area of V1. Uh, but anyway, like it's not my opinion. That's what I saw in the data. So whether you agree or not is another issue. But like
Okay.
my question is actually about the first slide that Antonia showed. On the second column, she showed this massive amount of veins on the top of the pile surface. Then they decrease across the postnatal uh, weeks or months. So how do you think this change affect your results, or do they affect your results? I kind of got confused about the time scales a bit, like whether this postnatal 0, 6 and 15-25 correspond to what you measured in your imaging.
So this was from an animal study, and this is kind of what they looked at vascular maturation. And the reason why I showed this is because I wanted to illustrate that we're not just dealing with the brain maturing and the cortex forming and these connections emerging and so on, but we also, especially when we talk about fMRI and the interpretation of it, it's important to mention that the microvasculature, which is relevant for 7T, is also changing. I think at the moment it's quite hard to tease that apart. That was something that Yucha, who I'm working with, her focus is on, because she wants to understand kind of how does the microvasculature mature? What is I guess, how much is it contributing to the bold response that we see and the changes in bold response that we see in neonates compared to adults? And she's tried doing that with modeling it. I mean, I don't know if Yusha, you want to talk about this, but yeah, it's definitely something that is contributing to the differences in the response that we see, and it's not just changes in neural maturation.
So related to this, just a quick follow-up. I know that there were very big contrast changes in neonates and babies. Like I don't know the details, but I remember it from your group's posters in the past. Um, does it also affect your percent signal change with regards to fMRI bold? Because if I mean, I'm just imagining like if T2 star contrast between white matter and gray matter is flipping, right, across months, and how it is interacting together with oxygenation related changes that you pick up with fMRI task activity. This could also contribute to some of the percent signal changes that you are picking up. Like, is this the case? I don't know whether the T2 star weighted contrast changes a lot in your um, baby brains.
Um, I don't know if should I answer the question first, or do you want to say something about the microvasculature?
I think they're kind of linked. So the lower vascular density actually within the cortex, I think means you have just a lower CBV within the cortex, and therefore you can have a lower change in CBV to neuronal activity when you have your bold response. But actually, if you take into account the T2 star and the more watery tissue properties, if you were to put that straight into like the balloon model, for example, your amplitude of response would be absolutely huge because of the differences in T2 star of the tissue compared with blood in the neonatal period. But then when you take into account differences in the vessel density, the blood flow, which is also much lower in neonates, then it brings the amplitude of the response right back down again. And if you look at 3T, the neonatal response has been shown to be like much smaller in percent signal change compared with adults, but that's not actually what we found at 7T. So I think at 7T, we might just have better spatial specificity and less partial voluming than at 3T, and therefore less kind of averaging out of the signal that we see. So actually, yeah, in neonates at 7T, the percent signal change is fairly compatible due to like the contrasting effects of the vascular density and the tissue T2 star. And the T2 star is very different from adults and neonates, but it's not changing that much in the short kind of window we're looking at. It inverts at 6 months. So the inversion of like the white matter, gray matter intensity occurs around 6 months.
Okay, thank you.
Maybe I have another question about the sensory device. And it's maybe more understanding question. So I understand you kind of you have the lower range, the lower speed, which is the CT range, and then when you're very fast, you're out of the CT range. But did you in the results you saw that the bold signal response magnitude was weaker when you have the fast speed, right? Is that so? Okay, I got that right. So I
Yeah.
guess your robot is kind of, it needs to accelerate very, very quickly to not be within the 0 to 10 regime and only be in the non-CT range and then stop very, very quickly again. So there's so that you're never really passing through the CT range when you're very fast. Is that so?
Yeah, so essentially, I mean, the robotics actually getting that into the correct range was a bit tricky because with pneumatic devices inherently are not super reliable and consistent because air pressure fluctuates and it's never perfectly sealed. So yeah, with a slower stimulation, we basically need to reach a certain threshold for it to then move. So the fast movement is always a bit faster, and then we can taper it down so it is reliably within that range that we would expect it to be. Yeah, but whereas the faster movement can kind of always be at this threshold and just yeah, move back and forth. When it comes to kind of the response amplitude, it was quite surprising that the faster stimulus had drove a lower response amplitude, especially because the RIs that we picked were overlapping. So we used the same RI in response to both stimuli because somatotopically they were aligned in terms of like the bold activation pattern. Yeah, why that's happening, we're not sure. It could be because with the CT fiber, since I mean the stimulus is a stroking stimulus, so it is kind of designed to be more tailored towards a CT stimulus rather than a non-CT stimulus. So it could be that that's why we see this much larger response amplitude, specifically in the superficial layers, which is kind of where you would expect CT inputs into S1 to go. Whereas kind of the A beta inputs into the middle layers, they, yeah, it's not as A beta specific because it is stroking instead of poking. So perhaps that's why we see a lower response amplitude despite the stimulation itself being stronger. But I'm not entirely sure.
Cool. Thanks. Do we have any more questions? Maybe a note to the folks joining here from the Martina side. Antonia will spend some months starting in January, right? In Boston at the Martina Center. So there will also be the chance to collaborate and ask many more questions as well. All right. With that, thanks a lot, and then see you in 2 weeks.
Thank you.